Hi guys, welcome to session 2. So this week is going to be all about cell division. Here is our virtual lab notebook, or our virtual notebook. Today, Monday, you're going to be working on 3.4, the cell cycle. Then Tuesday, 3.5, mitosis. Wednesday, meiosis. Thursday, cancer. So some of these things you might remember from 7th grade or have learned about, and that's great if it is review. If not, it'll be exciting, some new things. All right, so here's the PowerPoint for the cell cycle. So session 2, like I said, is all about cell division. Something fun in biology is that normally division means things are getting smaller, but with cells and cell division, it actually is an increase. So every time a cell divides, it is making more of itself. For example, here is one cell, first generation, and then it's going to divide and make 2 more cells. Each of those 2 cells is going to divide and make 2 more cells. So now in total we have 4. Each of those 4 cells is going to divide again and make 2 more. And now we have 8. So each generation, there's exponential growth and cells are multiplying while they are dividing. Okay, so here we go into the cell cycle. Before we do the Cornell notes, here are some questions to guide your thinking. So how many cells do we begin with? And when I say we, I mean every person on Earth. How many cells? 1, just 1. Okay, so here's a baby photo of you. This is a zygote. This is the moment after a sperm fertilizes an egg. The sperm burrows into the egg, fertilizes it, their DNA joins, mixes, and then creates you. So a zygote is the very first cell of an organism. Okay, so how do you get more? Started with 1 cell. How'd you get the trillions of cells that make up our bodies? Another question, how do little baby elephants become big elephants? Why do animals shed their skin? Why don't broken bones stay broken forever? And how are cancer cells different from healthy cells? So these are some questions we are going to answer this week. You'll be able to answer them confidently because you are going to learn about the cell cycle. Okay, so here is human life cycle, basically. So here's that zygote we were talking about. That's where we started. And this is where we are now, growing from that 1 cell into a baby, into a child, and then an adolescent, and then eventually adults. So here we are, this arrow, this is mitosis and development. All right, and here's the beginning of our Cornell notes. So key point number 1, the cell cycle. All new cells come from existing cells. You will remember this from when we learned about cell theory. Cells can only come from existing living cells. They don't pop out of nowhere, out of non-living things. They come from existing cells. The cell cycle occurs because cells are continuously growing and dividing. This is a constant cycle, meaning there is not a clear beginning and end. It flows into each other and there's not a start and stop. It just keeps happening. Cells growing, dividing, growing, dividing, growing, dividing. So the 2 main functions of the cell cycle, first of all, is growth, and second of all is replacement or repair. So for growth, that is when there is actually an increase in cells. It's important to note that cells are not growing and getting bigger. Cells are dividing and making more of themselves. So here's a little plant. Think about when it was a seed. It's growing bigger, not because its cells are growing bigger, but because its cells are dividing, making more cells. Here's an example of a human. There's the fertilized egg. It is growing and dividing. The cell is not growing. The cell is dividing, making more cells, and then the organism grows. Replacement or repair of old or damaged cells. That is what happens when there is an injury. So a broken bone, a cut, a burn, sun damage. anything where cells are replacing old cells. So even though there's an increase in cells, it's replacing cells that have died or are dying. So there's not really a net increase. Okay, an important part of the cell cycle is knowing about chromosomes. So chromosomes are tight bundles of DNA wrapped around proteins. So you already know about DNA. Here's the DNA structure. You're very familiar with the double helix, the A, T, G, and C. And think of this as a thread, like a teeny, teeny, tiny, not even microscopic. It's smaller than that. It's in the nano. And then it gets wrapped around these proteins, and that helps it be really, really tight and coiled and make this structure. So here's the DNA structure. It looks like an X. And here are the chromosomes. So they're drawn out here. We have 46 chromosomes. Humans have 46 chromosomes. And we have 2 sets of each. Where do you think we get each of those from? 1 from your mom, 1 from your dad of each. So overall, 23 chromosomes from your mom, 23 chromosomes from your dad. So overall, 46 chromosomes. And then each chromosome is going to have some unique genes on it. So chromosome number 1 has 246 million base pairs. That means 246 million A's, T's, G's, and C's. You can go to this cool website and you can see all of the different human chromosomes and the genes that are on them. So let's look at chromosome number 1. We can zoom in. And you'll see there's a bunch of names of diseases. And that is because the way that geneticists and doctors find out about which genes are on which chromosome is when there's disorders or problems or mutations, and they can track what went wrong in order to see what should have gone right. So if you look at all of these words, it's pretty much all diseases, different cancers, different things that will happen if any of these genes are mutated. And that's just chromosome 1. We can look at 7. Look at that. It's a little smaller. We can look at 14. A little smaller. Yeah, and I'll give you that website. You can play around with that. All right, back to our PowerPoint. Okay, key point number 2, stages of the cell cycle. So the cycle has stages. Here you can see it written out on the board. Today we are going to go over, in general, the 3 main stages, and then specifically what happens in interphase. Okay, so here is the stages of the cell cycle. And repeat these words after me. Interphase, so that's G1, S, and G2. And that's when the cell prepares to divide. Then mitosis includes PMET, and that's when the nucleus divides and splits up the replicated DNA. And then cytokinesis, that's when the cytoplasm divides, forming 2 new daughter cells. Remember, the cytoplasm is that jelly stuff that fills up cells. Okay, so zooming into interphase. So here we are, interphase, zooming into interphase. Gap 1, also called G1, is kind of boring. Cells just carry out their normal functions, they replicate their organelles, and they check their DNA. Here's a little picture, and we only really care about the DNA. So there's the DNA. The example is showing just 4 different chromosomes. It's all in the nucleus. And then out here are organelles that we don't really care about for right now. We just care about the DNA. Okay, synthesis. So that is the second step of interphase. And that's when the DNA is replicated. So remember, replica, make a copy. The DNA is doubled. And this is what you learned last week with the helicase and the DNA polymerase. That's when DNA makes a copy of itself. Okay, and then gap 2, that is another boring stage, and there is some additional growth and then a DNA checkpoint. So interphase is basically just to get ready to divide into 2 cells. The most the most important thing that happens is DNA synthesis when DNA is replicated. Something also about chromosomes, I said earlier that they look like X's. They only look like X's after replication. So here are 2 chromosomes, okay? And then here are the same 2 chromosomes after replication. Okay, now let's look at that in the human with the full set. Here are 46 human chromosomes. And here are 46 replicated chromosomes where a second copy has been made. And now it looks like an X because the copy that's been made is attached to the original. Okay, last key point, diploid versus haploid. So diploid, di means 2, so think 2. Diploid cells contain 2 complete sets of chromosomes, 1 from each parent. So as we said, human diploid cells have 46 chromosomes. An example of diploid cells is somatic cells, aka body cells, which is pretty much everything: your skin, brain, heart, digestive, muscle, everything, except for one thing you'll soon see. And the process that makes diploid cells is mitosis. All right, haploid cells. So human haploid cells have 23 chromosomes. Notice that that is half of diploid cells. So think of haploid, half, hap, hapa. Okay, so haploid cells have a single set of unpaired chromosomes, so half as many. And then an example, the only example, is reproductive cells. So sperm and egg. Those are what become the next generation. And those are made by the process of meiosis. All right, that is all for this. So what your little assignment is to go to the virtual notebook and then answer these questions. So these ones you're going to have to use your notes and your brain to think about it. These ones you're going to have to do some Googling, and it is just some quick searches, and it's about how many chromosomes are in different organisms and also the lifespan of certain cells. And after you do that, that is all for the cell cycle, and you can check those little boxes. All right, let me know if you have any questions.